Hello and welcome to Sam Biology class. Friends, today we shall discuss about the mechanism of prokaryotic transcription. Transcription is the synthesis of RNA from a DNA template. It is catalyzed by the enzyme DNA-dependent RNA polymerase. Transcription in prokaryotes is divided into a series of well-defined steps that are grouped into three phases. First phase is initiation, second phase elongation, and the third phase is termination. Initiation is the first phase of RNA synthesis. It involves three steps. First step is the binding of the RNA polymerase holoenzyme to a promoter region in the DNA. Second step is the localized unwinding of the DNA double helix around the transcription start site. And the third step is the formation of phosphodiester bonds between first few incoming ribonucleotides in the nascent RNA chain. In this figure, the upper strand is the template strand and the lower strand is the non-template strand. +1 represents the transcription start site from which RNA synthesis starts and proceeds in 5' to 3' direction. This is a promoter to which the RNA polymerase holoenzyme has to bind to initiate the transcription of a gene. Bacterial promoters have two conserved genetic elements. One is -10 element and the other is -35 element. This is the RNA polymerase holoenzyme, which consists of 6 subunits, out of which one is sigma. Sigma subunit direct the RNA polymerase to a promoter region. It is involved only in initiation. It has no role in elongation. The RNA polymerase holoenzyme initially binds to the DNA non-specifically and migrates along the molecule until the sigma subunit binds to the -35 and the -10 element. This binding results in the formation of a closed complex between the holoenzyme and the promoter. In such a complex, the two DNA strands are not yet separated. Hence, the name is closed promoter complex. The AT-rich -10 element then facilitates the localized unwinding of the DNA double helix around the transcription start site. This results in the formation of a transcription bubble in which the two DNA strands are separated. This is the transcription bubble. It is a molecular structure that consists of 12 to 14 base pairs. The localized separation of two DNA strands around the transcription start site results in the conversion of the closed promoter complex into an open promoter complex. In an open promoter complex, the two strands around the start site are separated. Hence, the name is open promoter complex. Now, the chain synthesis can be initiated. RNA synthesis is initiated by the incorporation of the first ribonucleotide into the transcription bubble. Because the nitrogenous base present at the transcription start site on the template strand is cytosine, So the first incoming ribonucleotide will be guanidine triphosphate. This is because guanine is complementary to cytosine. As a result, there will be base pairing between them. Then what is the second nitrogenous base on the template strand? It is guanine. So the incoming ribonucleotide will be cytidine triphosphate. Because guanine is complementary to cytosine, there will be base pairing between them. After base pairing, the first phosphodiester bond will be formed between guanidine triphosphate and the cytidine triphosphate. This reaction is coupled to the release of an inorganic pyrophosphate which undergoes subsequent hydrolysis to provide energy for this reaction. Next, what is the third nitrogenous base on the template strand? It is adenine. So the third incoming ribonucleotide will be uridine triphosphate. This is because uracil is complementary to adenine, so there will also be base pairing between them. After base pairing, the second phosphodiester bond is formed between these two ribonucleotides. In this way, a short RNA chain of 10 or more ribonucleotide is formed, which is then extended in the elongation phase. The incoming ribonucleotides of the nascent RNA chain must be complementary to the corresponding nucleotides of the template strand. Next phase of RNA synthesis is elongation. After initiation is over, the sigma subunit is dissociated and the core polymerase then extends the RNA chain. This transition from initiation to the elongation phase requires further conformational changes in the polymerase that enable it to grip the DNA more firmly. Like initiation, elongation of the RNA chain also takes place within the transcription bubble. During this phase, the RNA chain, which was produced during initiation, is extended by the addition of successive ribonucleotide to its 3' hydroxyl group. For the addition of each ribonucleotide, a phosphodiester bond is formed and an inorganic pyrophosphate is released. In this way, the RNA chain is extended in 5' to 3' direction. During elongation, in addition to RNA synthesis, the enzyme RNA polymerase performs an impressive range of tasks. Number 1, this enzyme has both DNA unwinding and rewinding abilities. This enzyme facilitates localized unwinding of the DNA double helix in front of the polymerization point and also facilitate rewinding of the complementary strands behind it. During elongation, the transcription bubble, which is a locally unwound segment of DNA, moves along the DNA. During this movement, there is localized unwinding of the DNA double helix in front of the polymerization point, and there is rewinding of the complementary strands behind the point. Number 2, this enzyme can also dissociate the growing RNA chain from the template strand as it moves along. And finally, RNA polymerase also perform proofreading. Next phase is termination, in which the transcription complex dissociates and the nascent RNA chain is released. It occurs when the RNA polymerase encounters a termination signal. The signals are specific sequences called as terminators. In E. coli, there are two types of terminators. One type that result in termination only in the presence of a protein called Rho is known as Rho-dependent terminator. Rho-dependent terminator. And the second type that terminate RNA synthesis without the involvement of Rho is known as Rho-independent terminator. Rho-independent terminators contain a GC-rich region followed by 6 or more AT base pairs with the adenines present in the template strand. The GC-rich region contains inverted repeats. When the GC-rich region is transcribed, the resultant RNA strand, because of self-complementarity between these two regions, fold upon itself to form a hairpin structure. This hairpin forms immediately. It retards the movement of RNA polymerase, thereby causing pauses in the chain extension. As adenine uracil base pairing is weak and less energy is required for the separation, the presence of a run of uracils after the hairpin facilitates the release of the transcript from the template strand. The second type of termination is Rho-dependent termination, that is similar to the Rho-independent process in that both of them involve the formation of the hairpin structure that impede the movement of the RNA polymerase. However, the Rho-dependent terminator contains two additional sequences. One is a 50 to 90 nucleotide pairs region located upstream from the inverted repeats. When transcribed, it produces the RNA strand which has more cytosines and few guanines. Such type of arrangement prevent the formation of hairpin or other types of secondary structure. The second additional sequence, which is located further upstream, specifies the Rho binding site known as ROTS. ROTS stand for Rho utilization site. Rho factor is a hexameric ring-shaped helicase. It binds to the ROT sequence and then it moves along the transcript in 5' to 3' direction. When the RNA polymerase encounters the hairpin, it pauses, allowing the Rho factor to catch up, which then passes through the hairpin and ultimately separates the DNA-RNA hybrid by its helicase activity. Friends, this is all about the mechanism of prokaryotic transcription. For more updates, subscribe my YouTube channel, Sam Biology Class.